Hello and welcome to the video overview of Chapter 3, which is all about carbon compounds and monomers and polymers that build up life. Um, Today we're going to be talking about how carbon, the atom, is used in four very different biological molecules: lipids, carbohydrates, proteins, and nucleic acids, to build really diverse sets of molecules that give us life. And today we're going to be talking about how carbon is integral to all of those different molecules and how we can understand basic structure equals function. The idea that the shape and structure of something determines its function, and when that structure is changed or modified for good or for bad, we see effects throughout different scales of life, whether it's cells or tissues or ecosystems. So the first thing we need to talk about is carbon, because carbon is the fundamental property for biological life and organic compounds. So anytime you see or hear something like organic life, it means that that living organism is structured around carbon-based molecules. So two things we have to ask to begin with is, why is carbon's atomic structure so critical for all of these diverse molecules we're talking about? And how does other important types of elements, nitrogen and phosphorus, come into the picture when we look at different types of monomers? So if you remember Chapter 2, where we were talking about atomic structure and valence electrons were those electrons in the outermost ring with the most potential energy, the least attraction to the nucleus. And elements that didn't have a full shell, generally 8, were reactive, because those electrons not only had a lot of energy, but they had a lot of space between different electrons, so they were volatile. Now, the reason why carbon is so fundamental for building all of these molecules we're talking about today is that it has the unique property of having 4 valence electrons, which means it can form 4 bonds, whether it's a triple bond and a single bond, 4 individual single bonds, double bonds. It's a lot more able to form lots of different bonds covalently with different elements, as opposed to nitrogen, which can only form 3 bonds, oxygen, which can only form 2, and hydrogen, which can only form 1. Now, one type of biological molecule that we're not going to be talking about discretely as a monomer or polymer today, but we'll see a lot more in future chapters, is ATP. And ATP is the energy currency of cells, meaning that when our cells move or breathe or replicate or create a protein, or if they're just doing normal daily life functions, they use ATP for energy. And the reason why ATP is so good at releasing energy is these 3 phosphate groups. So phosphorus itself is slightly polar as an element, but it's also bonded in each of these phosphate groups to an oxygen that has an additional electron. So all of these negatively charged atoms want to repel each other. There's a lot of negativity in this small space, which means when we indeed release ATP, we react it with water to break it apart, a lot of energy is released. So this sort of follows the form and function of the basic polymer-monomer reaction, where a big molecule reacting with water is going to lead to a smaller molecule and some sort of energy lost, and vice versa. If we're taking smaller molecules and building them up, we're going to need to take out water molecules and use energy to do that. So that brings us to the second concept of today, which is polymers and monomers. Monomers are simply repeating small molecules that, when covalently bound together, build up polymers. And the structure and function and form of a polymer, be it a lipid or a protein or a carbohydrate, really depends on what monomers are in that polymer and what's their arrangement and bond type. So we have to ask the question, how do monomers get formed and how are polymers broken down? How do these two things interact? And basically there's 2 basic types of reactions that we'll see with all of our four biological molecules. Dehydration synthesis or dehydration reaction is when we take water molecules, usually a hydrogen out of one molecule and a hydroxide molecule, OH, out of another molecule, and we form a covalent bond, creating a larger polymer and taking out a water molecule. Alternatively, let's say you ate a big bowl of pasta and you have a bunch of starch in your stomach. We need to use water molecules to break up those polymer molecules, leading to an OH being bound to now a free monomer and a hydrogen atom being bound to the polymer or another monomer. So hydrolysis is using water to break up big molecules. Dehydration is taking water molecules out to build up larger polymers. Now, the first type of biological molecule we're going to talk about are carbohydrates. Sometimes you'll see them referred to as saccharides. And the polymers that we see in carbohydrates are monosaccharides. Mono is the prefix for one, and polysaccharides are our polymers, poly being many. Three things we have to think about with carbohydrates. How does the structure and arrangement of glycosidic bonds influence the structure of polymers? Glycosidic being the specific type of bond between types of monomers, monosaccharides. How does the structure of different polysaccharides help explain their function? And why are branched carbohydrates so much more structurally sound and also less bioavailable for energy? So monosaccharides generally have the function of CH2O. So for example, glucose, which many of you have probably seen, is C6H12O6, where there's a 2-to-1 ratio between hydrogens, the carbon, and oxygens. Now, monosaccharides are often classified by having some carbon skeletal and having a C double bonded to an O at some point in their structure. So when we look at maybe 3 carbon, 5 carbon, or 6 carbon sugars, you'll note that one of the sort of conserved functional areas of each of these molecules is this C double O. Now, as I alluded to before, the specific type of bond that we see between monomers of carbohydrates is a glycosidic bond, where we're going to be taking a hydroxide molecule from one carbohydrate and another hydrogen from another carbohydrate, and that's going to result in a glycosidic link forming a disaccharide, such as sucrose, which is table sugar, which you might use in baking or coffees or whatnot. Now, polysaccharides are the polymers of sugars. This is where we have hundreds, if not thousands, of individual monosaccharides bound together. And as this whole chapter alludes to, the structure and function of these molecules are integrally linked and important. So 3 types of polysaccharides that you're going to come across in chapter 3, but also in future chapters and in the world, are starch molecules, often found in vegetable tissue, glycogen, which you will predominantly find in animal muscle cells, and finally cellulose, which we find in plant cells that predominantly give plant structure. Think about rigid plant matter such as bark or the stalks on a broccoli. And each of these, as you can see, are built up of glucose, but their arrangement is very different. Now, one type of polysaccharide that I alluded to is cellulose, and cellulose is what makes up a cell wall in plant cells. Now, while it is made up of glucose, it is a very different type of glycosidic linkage between those glucose cells, which not only gives it rigidity, but also it makes it not a viable energy source. So when we look at potatoes, for example, which are a universal carbohydrate food source across the world, it's really easy for amylase, which is an enzyme, to just cleave off these glucose molecules, providing energy for humans or animals. And the same thing is true with glycogen. While it is a bit more branched and a bit more compact for storage, it's really easy for our body cells to just individually break off these glucose molecules to maybe raise our blood sugar levels if we're in a soccer game or a running race or we haven't eaten for a few hours to give us quick available energy. However, you'll notice that cellulose is very branched and linear, and it has a different arrangement between those glucose molecules. So let's take a look. So when we look at starch molecules, it's a repeating 1,4 pattern between each glucose molecule. Now cellulose, on the other hand, is alternating. So we have dehydration synthesis occurring on an every other pattern, which makes it a lot harder to digest and break down with enzymes, but also it gives it a much stronger and rigid structure. So I'm sure you have all heard the saying, beans, beans, the magical fruit. I have to say no more. And part of that reason, whether it is humans or other organisms digesting cellulose, is that we lack the enzymes to break this up. And anaerobic bacteria in our guts are able to break these molecules apart, creating the second part of that saying. Now, lipids are the second group of molecules that we're going to be talking about. Lipids don't form true polymers because of their hydrophobic, water-fearing nature, but they do have some structural diversity that's really important to talk about within the context of life. The main types of lipids that we're going to talk about today are fats, phospholipids, and then steroid/hormonal molecules. To think about what we need to know with lipids are the differences between saturated and unsaturated fatty acids, how temperature influences the properties of lipids, and why phospholipids are a fat yet can have some affinity with water, which is really important for future discussions regarding cells. So when we look at fatty acids or fat molecules, there are two components that we have to talk about. The first is a fatty acid, which is just a carbon-hydrogen, we call these hydrocarbon chains, that contain carbon molecules with single bonds between each other and lots of hydrogen atoms bound to those carbon molecules. The second part of a fatty acid is just a glycerol molecule, which is going to be a carbon chain with some hydroxide molecules that allow us to form those ester links, which is the dehydration synthesis for fat molecules. So a triglyceride, or a fat molecule, is simply a glycerol with 1, 2, 3 fatty acids bound to it. And within those fatty acids, you can have differences between lengths, but also types of bonds within each of those fatty acids. What do I mean by that? Probably many of you have noticed that if you take a stick of butter out of the fridge, it doesn't melt unless you apply a lot of heat. It is stable as a physical solid at room temperature. Why is that? The 3 fatty acids in most saturated fatty acids contain carbon molecules saturated with hydrogen, meaning within this hydrocarbon chain, every single carbon has 2 hydrogen atoms. What this does is it creates really narrow, not space-filling fat molecules that are able to stay solid at room temperature because they don't have a lot of movement within their molecule. Alternatively, if you look in your pantry and you look at olive oil or canola oil or vegetable oil, they're liquid at room temperature, and the reason why is that they have what we call double bonds between carbons. They are unsaturated fatty acids. And when we take 2 carbon molecules and double bond them, we're taking hydrogen atoms out of the equation because carbon can only have 4 bonds per molecule. Now these kinks in these fatty acid tails create space between different fat molecules, which means they're more volatile to temperature and they remain liquid at 60, 70 degrees Fahrenheit. So the big difference between saturated and unsaturated fatty acids is there double bonds in the fatty acid tails? And are they stable or not stable at room temperature? Now, Fats are really good at energy storage. Per gram, you can get twice as many calories from fats than carbohydrates or proteins. So it's a really good way to not only provide energy, but store energy within organismal bodies. Now, another type of lipid that we have to talk about are phospholipids. Now, a phospholipid is a very similar structure to fatty acids. We have 2 fatty acids bound to a glycerol, but the difference is in that third carbon molecule, rather than having another hydroxide, we have a phosphate bound to it. And this phosphate, if you notice, has that phosphorus, which is polar, in this oxygen with a negative charge, which gives it a hydrophilic head. So when we look at cell membranes across humans or any other organism, most cells have a cell membrane comprised of 2 layers of phospholipids, where the hydrophobic tails point inwards because of their water-fearing affinity, while the hydrophilic heads, due to the phosphates, are able to interact with water. This is a really great way to separate internal and external environments from cells, but also regulate the movement of things in and out of cells. It basically creates a castle wall of sorts for cells. Now, the last type of lipids we have to talk about are steroids. Now, steroids are lipids that do have a carbon skeleton, and they have some level of structure that provides a really specific function when it comes to their ability to influence living organisms. One type of steroid that we often talk about in the world of human health is cholesterol. Cholesterol is a really important steroid that is actually embedded within cell membranes, and they actually space out these phospholipids, providing a membrane that is fluid. However, there are other hormones like estrogen or testosterone that are important hormonal molecules that are able to travel throughout the body and influence cells and tissues by going through these cell membranes and directly targeting genes, such as maybe bolstering the amount of growth hormones in cells in adolescence as you grow and develop muscularly and skeletally. And here's a good example of what a steroid molecule looks like. We have these 4 carbon rings, and then some additional functional groups that provide structure and function to them. So testosterone and estrogen, we often classify those as biologically male and biologically female. Sex hormones are very similar, the difference being what's bound to these 4 carbon skeletons. Now, the last 2 molecules that we're going to talk about are a little bit more complicated because they are so diverse within their structure and order and function: proteins and nucleic acids. So when we talk about proteins, we have to think about what are the components of amino acids that form peptide bonds. When I'm looking at a polypeptide, how does the change in one amino acid change the function and shape of that protein? And how does the structure of completed proteins enable diverse groups of functional proteins, which there are many? So proteins, if we took all the water out of your body, account for about 50% of your cells. So proteins are super important, not only building up living life, but they also have a whole host of diverse functions when it comes to things that they do for your body. So there are 8 that we're going to briefly talk about with specific examples, just to give you some more awareness as we move throughout AP biology and talk about these different groups. One specific type of protein group are enzymes. Enzymes are super important for breaking down molecules or building them up. Some of you might be lactose intolerant because you don't have enough lactase in your bodies to digest lactose. Defense proteins are super important for secreting or building antibodies that can specifically target and neutralize viruses and bacteria. So if you've gotten a COVID-19 vaccine, your body is now able to identify COVID-19 and produce antibodies, neutralizing the infection or at least minimizing the risk of it. Storage proteins are really important for a variety of reasons, especially when it comes to the development of embryos. So if you crack open an egg, the egg white, which is known as all oval albumin, is not only a delicious food source that can make things like meringues, but actually when it comes to the chicken developing in the egg, those amino acids are really important for that embryo to develop and obtain energy from. Transport proteins, which we find in those cell membranes that we talked about back with lipids, are super important for getting things in and out of cells. For example, hemoglobin, which we find in red blood cells, has specific proteins that allow oxygen to travel in or out of the red blood cells, depending on if we're bringing oxygen to the cells or if oxygen is leaving to go into the cells. It's a really important protein. Without it, we wouldn't be able to live. Hormonal proteins, well, some hormones are lipids, like estrogen and testosterone. Other hormones are protein-based. So maybe you might know someone that's diabetic or has some insulin sensitivity issues when it comes to a lot of sugar. Insulin is a protein that is secreted in the body to help normalize and regulate blood sugar, whether we just ate a lot of Skittles or maybe we haven't eaten in a while and we need to get our blood sugar levels up. Receptor proteins, maybe some of you play a sport where you've experienced what's known as a runner's high. Receptor proteins are super important in neurons to receive and respond to different types of signaling molecules, one of which is dopamine, which is a really feel-good molecule that is elicited in a variety of experiences, whether you're eating chocolate or going on a long run. It changes your emotional state of being based on these receptor proteins being responsive to dopamine being released. And then contractile and motor proteins are really important for your body to be able to move. So every time you contract your bicep doing a barbell at the gym or extending your quadricep to kick a ball in soccer, there are strands of actin and myosin in your skeletal muscles that are contracting and expanding, allowing you to have that movement. And then there's also structural proteins. So many of you have probably heard about scurvy, which was a common ailment that many sailors had in the 16th and 17th century. And that was due to the breakdown of collagen, which is a really important protein that actually holds your teeth in and provides structure to your fingernails. So a common ailment of scurvy or a vitamin C deficiency was this collagen breaking out. And as a result, people would have their teeth fall out and their fingernails falling out because that collagen didn't have the appropriate structure. So lots of different proteins in the world. How are they built and what gives them all those unique structures and functions and properties? Now, the fascinating thing is that polypeptides are all built from the same set of 20 amino acids. And as those amino acids are arranged and built, a protein becomes a functionally specific molecule based on their order and the interactions of those amino acids. Within one amino acid, we have three groups that are really important. We have the R group, more on that in a second. We have the amino group, which is a nitrogen with two hydrogens. And we have our carboxyl group, which is a C double bonded to an O and an OH. So when we form polypeptides, these two groups are going to be where we see the peptide bonds forming. Now, when it comes to amino acids, we have four flavors, so to speak. The first would be hydrophobic or water-fearing amino acids based on their R groups. Notice that all of these have pretty non-polar molecules, lots of carbons with hydrogens, not a lot of oxygens in these R groups, so they're going to be water-fearing. Alternatively, we have hydrophilic or water-loving R groups. Notice that we have some oxygens and some nitrogens, so atoms that tend to be a little bit more polar in their nature when it comes to elemental sharing. And then we can have negatively charged or positively charged molecules where atoms either have gained or lost electrons, giving them a slightly different flavor than just the water-loving or water-fearing R groups. Now, when we form a polypeptide bond, we are going to be taking the hydroxide group from that carboxyl acid group and taking out a hydrogen from the amine group, forming a water molecule. So there's another good example of dehydration synthesis. Now, here is a space-filling model of a protein from a flu virus and an antibody protein. And notice that we have a really specific matchup between the antibody protein, the thing created by our immune system to neutralize the flu virus, and the flu virus itself. So proteins have really specific structural groupings based on how these amino acids come together. So when proteins are created, the first thing that we look at in terms of structure is primary structure. And that's just the beads along a string sort of mentality, where amino acids are going to be forming peptide bonds, and we really don't have any specific functional groupings yet. There's no shape or function to the peptide. Now, as the protein is created, we start to see folds, whether it is an alpha helix or maybe some pleats within the protein, and those are going to be forming just based on weak associations of hydrogen bonds between negatively and positively charged groups. They're not R group interactions yet. It's just the fact that as our protein is being created, there's going to be some negatively charged areas and some positively charged areas. And based on electromagnetic attraction, they're going to start interacting with each other. Then we get to tertiary structures where we start to see some really complicated folds occurring. And the tertiary structure is going to be a result of those R groups starting to form. So we might see an inward bend where we have a lot of hydrophobic molecules. And then we might have some less bendy regions where we have hydrophilic amino acids because they're able to interact with the water. Quaternary structure is where we have multiple tertiary proteins come together forming some biologically diverse molecule. And this right here is a red blood cell where we have 4 hemoglobin molecules, 2A and 2B subunits, coming together to form a structurally complete protein and cell. And this is where we can start to see the different interactions forming protein structure, whether it be hydrogen bonds or weak interactions based on the presence of electrons. We might have a disulfide bridge occur between 2 amino acids. We might have that positively and negatively charged R group starting to create the protein bends and shapes. Now, as I alluded to earlier, hemoglobin is a super important protein and cell when it comes to biological life. And while it is important to have a fully functional and formed protein, that doesn't always occur. So when it comes to the creation of proteins, the order of amino acids is super important. So on the top of this figure, we have a normalized red blood cell where we have 7 amino acids in order that form a beta subunit, which comes together to make normal hemoglobin. And we don't have those red blood cells clumping, and they're able to carry oxygen. But in a lot of people, especially in regions in malaria-prone countries, sickle cell anemia tends to be an affliction based on one change in amino acid. And the glutamine in position 6 is replaced by a valine. Now, this is based on genetics. It's not a random occurrence, but that one amino acid being switched out causes a new hydrophobic region to occur in that beta subunit. And because that normalized hemoglobin that usually interacts with blood and water easily is now hydrophobic, they all start to clump. And their shape is a lot different based on that one amino acid substitution. And that means that the ability to carry oxygen is significantly reduced. So it's not as if you have sickle cell anemia, you can go out and sprint as fast as you can. Your body's ability to carry oxygen is significantly hindered because of this one small substitution. Now, there's a variety of things that can also influence protein structure, whether it's pH, salt, temperature, or other environmental factors. When a protein is put in an environment that it's not normally used to, it is denatured. The best example I can give you is you crack open an egg. The albumin, which is the egg white, is liquid, it's very fluid, it's viscous. You can put in a bunch of things to give it structure, but as soon as you heat it up, that egg white solidifies. And that's where the change in temperature is causing that protein shape and structure to unravel. And we can have lots of normalized proteins be denatured based on one of those environmental factors. That's why it's really important if you have a fever over maybe 102 Fahrenheit that you seek medical help, because your body is at such a temperature that many of the proteins in your body are getting cooked like an egg white on a skillet. Not necessarily the most safe thing. Now, the last molecule, I know we've been here for a while, are nucleic acids. And this is the programming for our amino acid sequence. So when we think about proteins and we think about polypeptides, their sequence order and inherent structure and function is based off nucleotides in their order in what we call a gene. And genes are made up of DNA, which is a type of nucleic acid, the monomer being called nucleotides. So 3 things we have to think about with DNA or nucleic acids. What is the backbone of DNA and RNA, and how does this form nucleoside or nucleotide structure? Which nucleotides are found in DNA and RNA? What's the difference between a purine and a pyrimidine? And how does the shape and bond between nucleotides prevent errors in the DNA code? Because like a computer, errors in the code causes a whole host of problems. So there's 2 types of DNA that we need to talk about, or I should say nucleic acid. There is DNA and then there's RNA. DNA is really important for genes and for proteins, and RNA tends to be more of the middleman between DNA and amino acids. And there's some very important structural differences between these 2. When we talk about the gene expression in protein creation, DNA, which is found in the nucleus of a cell, is going to be coded and expressed, leading to mRNA being produced. That mRNA is going to leave the nucleus, go into the cytoplasm or the main body of the cell, and then it's going to be using ribosomal subunits, which are a type of protein, to create our polypeptide. And as that polypeptide is formed, its structure and function is going to be created. Now, what the heck do we need to know about the form and function of the monomers with nucleotides? Nucleotides are the most complex molecule when it comes to their monomer structure. We have 3 specific parts. We have a nitrogenous base, we have a 5-carbon sugar, which is known as a pentose, and then we have a phosphate group. Phosphate, again, is a conserved functional group that we've now seen in a couple different types of molecules. Now, there are 5 flavors or monomers of nucleic acids. We have pyrimidines and we have purines. Pyrimidines are going to have 1 nitrogenous ring attached to the carbon sugar. We find cytosine and thymine in DNA, and in RNA, uracil replaces thymine. Now, purines are the other 2 nucleotides that we see in both DNA and RNA, but they have 2 nitrogenous bases, adenine and guanine. A word of wisdom that I learned in 2010 in my AP Bio class for remembering the difference is purines make me gag. So because purines have the double ring, they're bigger. Think about trying to get too much food down your throat at one time. You gag, guanine and adenine. Now, the difference between DNA and RNA is what do we see in that 5-carbon sugar. In deoxyribose or DNA, we have one oxygen missing. In RNA, there is an oxygen present bound to that second carbon. The key difference in terms of the functionality of that difference is that when we remove the oxygen from this 5-carbon sugar and see it in DNA, it's a lot more shelf stable. Oxygen, again, is a very volatile electronegative atom. We want our DNA to be stable. And the less oxygens we have in that molecule, the better. RNA really doesn't have a long shelf life. So that oxygen there is part of its functionality of not being a long-term conserved molecule. Now, the last thing we need to look at when we look at the structure of a nucleotide is that between each nucleotide, a molecule is going to be bound between the 5-carbon and 3-carbon sugar of each nucleotide. So we're going to have that phosphate with an oxygen bind to an OH group off the 3-carbon sugar. So we always say that DNA goes from 5' to 3', from the 5-carbon sugar to the 3-carbon sugar. So if we wanted to add another nucleotide, we would be binding it to that OH group right there with this phosphate. Now, sugar phosphate backbones, which I just alluded to, are the main structural group that creates DNA. And we see that DNA is antiparallel because one strand always travels 5' to 3', and the other side goes from 5' to 3' in the opposite direction. Thymine and adenine are the two nucleotides that bind together, while guanine and cytosine always bind together. And they do this based on differences in the number of hydrogen bonds that they can form, but also the differences between pyrimidines and purines. So if you notice, 2 plus 1 is 3, 1 plus 2 is 3. If we had two purines try to bind together, the railroad tracks, so to speak, of DNA would be too wide. It wouldn't work. And if we had two purines try to bind together, forming this double bond, the DNA would be too narrow. So by default, using a purine with a pyrimidine, we're able to create a molecule that has some built-in properties to prevent errors as it is replicated. I hope you have been able to make it through this video. Again, if you have questions, email me, and I will see you in class soon.